biochem students. We're going to continue where we left off and try to get through fatty acid synthesis. I probably won't finish the slides, but we'll finish them on Tuesday, but this will allow you to get through the main component of fatty acid synthesis so you can be studying that for the quiz. Okay, so this is slide 29. We ended on slide 28. And the last thing that we needed to look at on beta oxidation before we look at fatty acid synthesis is what happens if you have cis versus trans bonds. Okay, so if there is a cis bond, in regular beta oxidation, the double bond is trans, but if there's a cis bond, for example, in this case, it's circled in red, then all that has to happen is an isomerase converts that cis to trans, which you can see here. That's all that you need to know from this slide, is that in unsaturated fats, we convert from cis to trans using an isomerase. Okay? So now I want to talk about fatty acid biosynthesis, because that's the other huge component of Chapter 12. We'll finish the later parts of Chapter 12 on Tuesday. But in fatty acid biosynthesis, the main things, there's a lot of differences here. It's a different enzyme. The enzyme is fatty acid synthase, nice and easy to remember. It takes place in the cytosol, whereas degradation, the beta oxidation degradation, was in the mitochondrial matrix. It's going to use ATP, but it's going to differ. The number's going to differ depending on the chain length. If you remember, for fatty acid biosynthesis, for fatty acid degradation, 2 ATP were used. That was on your quiz to activate at the beginning. But other than that, the ATP equivalents we just calculated at the end based on the chain length. And in this case, we're going to see a different number of ATPs as well. Okay, so you need to pay attention to that. And there'll be a summary slide as well with some of the differences. Okay, the enzyme is a huge enzyme. You can see several structures of it on the right there. It's a large, lots of different cofactors, lots of components. So fatty acid synthase is large. One of the main differences is it uses a different cofactor. In this case, the cofactor is the acyl carrier protein, or ACP. Okay, so on the fatty acid synthase, this cofactor, if you look, I have a kind of a circle here, that'll be the large protein, and then one of the amino acids, serine, contains the cofactor acyl carrier protein, which is shown in green. If you look, underneath it is coenzyme A, which we've seen before. The green component is identical between the acyl carrier protein and the coenzyme A. If you remember when we were talking about coenzyme A, the compound, where the compounds bind to the coenzyme, it's going to be on the sulfur group. It's going to be the same thing on the acyl carrier protein. The sulfur on the end is where your particular fat or whatever we're binding to, the ACP is going to bind. It's going to be on that end. But big difference. Breakdown uses coenzyme A, although we will see it a little bit in some of the fatty acid biosynthesis. The fatty acid biosynthesis has the acyl carrier protein on the fatty acid synthase. Okay? So let's look at the net reaction just for palmitate. If you remember, palmitate is a 16 carbon fatty acid. So it's going to take 8 acetyl-CoAs, 8 acetyl groups. Remember, acetyl group is a 2 carbon unit. You need to know this net reaction, and as we go through the steps, it'll be clear where each of the appropriate, NADPH and stuff is used. But here's just the net reaction to start with, okay? We'll start with acetyl-CoA, which once again normally lives in the matrix. It's going to get smuggled out of the matrix into the cytosol, because remember, this is taking place in the cytosol. It's going to get smuggled out as citrate. So here's your citrate in the matrix, your acetyl-CoA. Citrate on the right, so we smuggle it out into the cytosol. And then on the right here, there's your acetyl-CoA is going to get used in lipid biosynthesis, where you've seen that before in this class, where it just says acetyl-CoA to lipid biosynthesis. Well, we're going to go through the steps today, okay? So when the citrate levels are high, or the energy requirements are low, then the citrate is going to get converted into oxaloacetate, leaving us with acetyl-CoA that's ready and available to use for fatty acid biosynthesis, okay? So the next slide is going to start upper left here with the acetyl-CoA group. So in the upper left, there's your acetyl-CoA from the previous slide. That red 2-carbon unit, that is the unit that's going to be the growing chain to form whatever the new fatty acid is that we're forming. Okay? On the right, we have a new group called malonyl-CoA that will become malonyl ACP. So that malonyl group will get added onto that acyl carrier protein, that ACP. Both of these sides occur simultaneously at the beginning of fatty acid biosynthesis. So I'm going to label them as steps 1 and steps 2. So both step 1, step 2. We're going to have this 2-carbon unit get added onto the fatty acid synthase, and you're going to see acetyl synthase and malonyl ACP. Okay, so both of those go together. Something to note with the malonyl is the coefficient for malonyl and the ATP is going to match, because it's going to be for every 1 malonyl, it will be 1 ACP. Or excuse me, 1 ATP used. The other steps, which like I said, we'll go through them step by step. I've numbered them here, 3, 4, 5, 6, 7, and then I just call the last step end, because it's not necessarily step 8. We will repeat until you have the appropriate fatty acid length on the growing chain that is being synthesized on this huge fatty acid synthase complex. Okay? So the top part that's shaped kind of like a Y, that'll be steps 1 and 2. We're going to see that on the next slide, and then we'll go through each of the 3 through 7. We'll go through each of the steps individually. Okay, so this is the beginning. We have this blob on the bottom is your fatty acid synthase. This is reactions 1 and 2 loading the enzyme. Remember, we have our acyl carrier protein with the appropriate group. There's also a cysteine group that we're going to see with its side chain. We're going to add the appropriate acetyl group and malonyl group onto the 2 components on the fatty acid synthase. So if you look, the acetyl group got added onto the cysteine, and the malonyl group got added onto the acyl carrier protein that's on the right. Okay, so both of those groups are now loaded onto the enzyme. The blob is, once again, the fatty acid synthase enzyme. Both of those are there. Now we can do chemistry between the 2 of them to grow our fatty acid chain. Okay? So that's steps 1 and 2, and I just call it loading the enzyme. We get malonyl-CoA on the enzyme. We get acetyl-CoA on the enzyme. Now I won't make you draw this, but you will have to recognize them. Okay? So what's on the left here is what was on the right on the previous slide. So we're starting with doing some more chemistry here. Step 3. It's a condensation reaction. So we're going to decarboxylate that malonyl. So that CO2 is leaving and allowing the 2-carbon group that's on the cysteine to get transferred over, giving us a 4-carbon product. Okay, so the 2-carbon fragment on the cysteine is going to attack the acetyl part on the ACP, giving us this 4-carbon product. Okay, so you need to know that it's a condensation reaction. You see CO2 is leaving, 4-carbon product. Moving on to step 4. It's a reduction reaction. So this is where the NADPH that you see in the net reaction, one of the NADPHs is going to get used here in step 4. So you can draw the NADPH that way, or you can do what we usually do. NADPH, etc., etc. You can do it that way if you prefer, and then NADP plus one. NADP plus. You can do it that way. It doesn't matter. Okay? But what you can see in the reduction is that this ketoacyl group is getting reduced to form the OH group. So that's getting reduced in step 4. Still the same 4-carbon product. Step 5 is a dehydration, so we're going to produce a double bond there between the 2nd and 3rd carbons. Excuse me. That's it. That's just step 5 is a dehydration, and step 6 is another reduction. That's why you see so many NADPHs in the net reaction, because step 4 and step 6 both use NADPH. So in this case, NADPH, double bond is reduced again. We have a 4-carbon group that can go on to step 7. Okay? So it's a complete new acyl group. that can get moved, it's going to get moved in step 7 over to the cysteine so that we can continue to grow with it by introducing a new malonyl-CoA. So in this point, we finish the chemistry of what used to be the acetyl group and used to be the malonyl group. And go to step 7. Okay? Step 7, a new malonyl-CoA comes in, so CoA has to leave. And this group got moved over onto the cysteine. And then the malonyl group got added onto the ACP. Okay? Just so you know, this CoAsh at the bottom here, that's not in the figure in your book. I added it in my book because I am, in my figure, adding a new malonyl group onto the ACP, that acyl carrier protein. And this just repeats over and over until you have the appropriate length of fatty acid. And then the thioester gets hydrolyzed. It gets hydrolyzed on the thioester end. That's important to note. The ACP is attached at that particular end. This fatty end, or the acid end. That's the same thing that we saw with fatty acid beta oxidation, is we're dealing with that end, not the end that's the long tail. We're dealing here where the arrow is. We're not dealing down there. But we're at the top of the tail. Okay? This end, you may say, is part of this hydrolysis, is part of step 7. It doesn't matter. I won't ask you the number. I won't ask you the number. Okay, so now that we've gone through it step by step, look at the net reaction again. There were 7 malonyl-CoAs used, therefore 7 ATP are used. The only way to know that is to know the math for the acetyl-CoA and the palmitate. You have to go through it 7 times. There's 14 NADPHs because there's 2 used per round. So total, if you were to look at the ATP, there's 42 ATP used to make palmitate. Whereas if we were breaking down palmitate like we did in class, we actually, if you remember, calculated 106 ATP from that. So it's using less ATP to make than what palmitate actually provides for you. Now, if you didn't like the figures that I provided for you, there's another overview that I have here that shows the chemistry of the 2 groups on the top left. In this case, the green is the acetyl-CoA, and then the malonyl group that gets decarboxylated. If you look, the CO2 group is in blue, and you can follow the addition of the 2 carbon units on this. You have this figure, like it's 3 slides. Okay, if you want to see through step by step by step, if that helps you as a review, be my guest and look at those slides. So when we look at metabolism, the only other thing that we need to look at is regulation. So look at this figure on the right. If you look, I'm going to go, we got the plasma membrane at the top, and down here we're in the mitochondria. There's the acetyl-CoA made from beta oxidation that we already saw last class. There's the acetyl-CoA. If you look, up here is the citrate, and then there's your fatty acid synthesis out in the cytosol. Okay? There's your carnitine shuttle that we already looked at last class, and then they have little green circles and red dots to show you, hey, guess what? Citrate will activate fatty acid synthesis. Palmitoyl-CoA, however, will inhibit fatty acid synthesis. Malonyl-CoA inhibits this carnitine transporter that we saw last class, and that's important to note because the concentration of malonyl-CoA in the cell is going to prevent simultaneous fatty acid synthesis and beta oxidation. You don't want them both to occur together, so we need to have a regulator. Malonyl-CoA is that regulator. Okay? So you need to know that we can stimulate by citrate, we can inhibit the fatty acid synthesis, hopefully that makes sense, with fatty acids, and citrate will activate. So hopefully that makes sense. The carnitine transporter that I on the board, here's a figure for it. If you want to look at the structures, you have this figure. So that's the transporter that's on the bottom right of the previous slide. Here's another figure showing regulation, but this one shows beta oxidation out here, and then fatty acid breakdown, fatty acid synthesis out here. So on the left is the matrix, the right is the cytosol, and the cool thing is it shows The citrate, it's labeled transporter in this figure. Transporter shuttle, transmembrane protein, it doesn't matter. And then the carnitine shuttle down here. So you